Students, welcome to our next session. In previous class, we discussed about the production of humulin. Continuing the second topic in major heading, that is application of biotechnology in medicine. First topic was insulin or humulin that we have done, and today's topic is gene therapy. Very important topic. Right? Gene therapy. As the name suggests, right? Gene therapy, you know what is therapy means? The process of treatment. The process of treatment is what we call as therapy. So here, as it says, gene therapy means by using genes, by working at the genetic level, we cure diseases, disorders. That is what is known as gene therapy. In your genetics chapter, you might have studied many disorders. In fact, genetic disorders, you have studied Y chromosomal disorders, X chromosomal disorders, recessive disorders, dominant gene disorders. So many genetic disorders are there. So far, it is been detected in human beings. So far, around 4,000. How many? 4,000 genetic disorders have been detected. Most of the genetic disorders cannot be cured. This is a point also you might have heard, but there is an answer for that. Is this one? Genetic disorders can be cured, can be treated. Certain, not all I am saying. Certain genetic disorders can be cured. When it can be cured? At the embryonic level. This is very important factor. Gene therapy is done at the very early stage. At the very early stage, or in the infant stage, or the best treatment is at the embryonic stage, when the fetus is developing when the fetus is developing, or when the embryo is in the mother uterus, developing stages, developing condition, or very immediately after birth, right? So this therapy has to be done. Such a therapy is what is known as gene therapy. Gene therapy. This is the introduction to the background of the gene therapy, the historical background. Now, going on to the definition, gene therapy is defined as the collection of methods of correcting the gene defect in the embryo is what is known as gene therapy. The collection of methods of correcting the gene defect in the embryonic stages or in the embryo is known as gene therapy. This is the definition given. For understanding, we can say that the therapy that involves the replacement of the defective gene. Replacement means the defective gene is removed, in place of that the functional gene, the normal gene, the functional gene is placed. Fine. Defective gene is removed, deletion of the defective gene, addition of the correct gene, normal gene, functional gene at the embryonic level is what is known as gene therapy. What is known as gene therapy. This gene therapy was demonstrated in the year 1990 by William Anderson French. Some book says William French Anderson. Well, William Anderson French I have studied. Some book says William French Anderson, whatever it is, proper noun. Improperly you can say. That's what is proper noun. Okay. Anyway, so these are the scientists who first demonstrated. He demonstrated how it has to be done in one way, but later now the gene therapy has been modified, has been improved. But he basically the basic method of the idea of the concept of the phenomena of gene therapy was given by William Anderson French in the year 1990 by treating a syndrome called severe combined immunodeficiency, what we call SID, SCID, deficiency, severe combined immunodeficiency syndrome. It's a syndrome. You know what I mean by syndrome? Many times I would have told you the many problems or many symptoms, many defects at the same time is is what is collectively known as syndrome. This is severe, very serious, chronic, severe case. Combined, why combined here? We say because of both T lymphocytes and B lymphocytes. Just recall a bit of 8th chapter, immune system immunity. What is the concept? What is the contribution of B lymphocytes and T lymphocytes? How they are being produced from bone marrow. Fine. So here combined because 2 lymphocytes and immuno. Why immuno? Because immuno. What it says? Immunodeficiency means what? The immunity is decreased, deficient, less than the normal. Deficient than the required, minimum than the optimum, less, decreased, reduced. That's why we call it as severe combined immunodeficiency syndrome. How this occurs? Why does this occurs means there is an enzyme known as there is a gene known as ADA gene. Now what is ADA stands for? Adenosine deaminase. Adenosine deaminase gene. This is a gene that is found present on 20th chromosome. Recall your genetic chapter, molecular biology chapter also. I have told you many times, to understand biotechnology, you need to have a solid knowledge, basics knowledge of molecular biology, genetics and microbiology. So immunodeficiency. Why this deficiency occurs? How this syndrome occurs means to say that there is an enzyme, there is a gene known as ADA gene, adenosine deaminase gene. What is the function of this gene to produce adenosine deaminase enzyme? What is this? It produces a enzyme known as adenosine deaminase. This enzyme is produced when this gene is present on 20th chromosome. Normally in all normal human beings, this gene is found present on 20th chromosome, the largest chromosome, 20th chromosome. This gene is present over here. The function of this gene is to produce this protein, adenosine deaminase, considered as enzyme, basically a protein. You all know one gene, one protein concept. When this enzyme is produced, what happens here? The B lymphocytes from bone marrow and T lymphocytes from thymus gland, immunology chapter, both these gets activated and they perform the normal function. When they perform normal function, that person has the correct immunity. When they are not produced, or when this gene is absent, this enzyme is not produced. When this enzyme is not produced, these lymphocytes are not functional. Because of this reason, there is a decreased immunoreaction, there is a decreased immunity, there is a less reduced immune response. Such a disorder, such a syndrome is what is known as severe combined immunodeficiency syndrome. How this occurs? Because of the lack of ADA gene on the 20th chromosome. What is the function of ADA gene? To produce a adenosine deaminase enzyme. When this protein and this enzyme is produced in the body, it stimulates the B lymphocytes and T lymphocytes to produce specific antibodies. Because of the lack of this gene on this chromosome, this whole program, this whole process does not take place in the body and results into the SID. So this disease was cured by, treated by earlier enzyme replacement therapy. This is not there in your syllabus chapter, but to understand the background, to treat this earlier, enzyme replacement therapy was done. Bone marrow replacement was done. That is also done in cancer also. Blood transfusion was done. Many other techniques were tried, but they are not so accurate. They were not so curable, and moreover very tedious process, lengthy process, and patient has to do it regularly on a recurring basis at regular intervals of time, repeatedly. That has to be done. Bone marrow replacement, blood transfusion, lymphocytes has to be given, enzyme replacement therapy, repeatedly it has to be done in the whole lifetime. So for that, to overcome all these problems and issues, in the year 1990, and William Anderson French demonstrated how the gene therapy can be done. Now the simple basic procedure of gene therapy, how it is being done, means to say that in the very first step to cure, we will take a classical example of this disorder only, this syndrome only, that is SID. It can be asked in NEET also, right? Which is the syndrome? Who cured? What is the deficiency enzyme? Which is the gene? Where this gene is found present? All these NEET questions we will cover. So here, how to demonstrate it? What he took was, he took the blood from the patient, and he demonstrated this on a 10-year-old girl for the first time. He demonstrated this on a 10-year-old young girl. English is a funny language, right? 10 years old, young, two very opposite words I am putting it here, just to make it more interesting. 10-year-old young girl by name Ashanti De Silva. Ashanti De Silva. See the name of this girl, pretty girl. Ashanti, opposite of Shanti. She was suffering from this disease, this syndrome. Which syndrome? SID. She was this girl, Ashanti De Silva, was suffering from a severe syndrome, severe combined immunodeficiency syndrome, SID. This and William Anderson friend for the first time cured this girl by using gene therapy methodology. How? See the procedure. Blood of the patient is taken, removed, not completely, partly. Right? Blood of the patient is taken. From this blood, right? This blood, lymphocytes are extracted. You all know T lymphocytes, B lymphocytes will be circulating in the blood. Lymphocytes are extracted. Then these lymphocytes are grown. How? They are cells, they are sites, they need a nutrient media. Blood is also very rich in nutrients. So cells will multiply in blood. Which cells? Bacterial cells, other cells during infection, during diseases. Similarly, these lymphocytes also proliferate. So here they are grown outside in the laboratory condition, in vitro condition, laboratory condition. They are grown by using media. So in these lymphocytes, the normal is attracted from other person, this gene, this enzyme is injected. What is injected? The third procedure. The enzyme, ADA enzyme, adenosine deaminase is injected into the lymphocytes. Which lymphocytes? B lymphocytes, T lymphocytes. Because these lymphocytes lack that, the blood lacks this enzyme. Because of the lack of the enzyme in the blood in the body, these cells, B lymphocytes and T lymphocytes, are not performing their normal function. They are not increasing the immunity. To overcome this problem, this enzyme has to be, ADA enzyme has to be injected into the lymphocytes. After that, in the next step, this healthy lymphocyte. Why I am calling this as healthy lymphocytes now? Because they possess what? ADA enzyme. This healthy lymphocyte, along with the blood, along with the enzyme, has to be given back to the patient. Injected back to the patient. So by this way, this therapy was being demonstrated. It is a very simple, basic procedure of gene therapy. When the gene therapy is actually done, fine, at the genetic level, this is not how it is being done. It is done at the genetic level. From the embryo, from the amniotic fluid, from the embryo, genes are taken, the cells are taken. You have studied HGP, Human Genome Project, in your molecular biology chapter. That is known as biological moonshot, because it is such a big achievement in biology, in biotechnology. Hence it is known as biological moonshot. You might have studied that in molecular biology chapter. So here, Human Genome Project, we can see that which gene is found present and where, what is the function. So that gene has to be replaced. That's what is exactly gene therapy. But the gene therapy concept was demonstrated by William Anderson friend. He did it like this. So after that girl was being cured. Earlier I told you, enzyme replacement therapy was also done to that girl. Now there are two types of gene therapy. One is known as germline gene therapy. Germline, if the gene therapy is done at the germinal cells. What do you mean by germinal cells? The gametes, fine. In the X chromosome, Y chromosome genes defective, if gene therapy is done by replacing those genes, such a therapy is known as germline gene therapy. Other is known as, very simple, somatic. This word you might have heard many a times. Somatic cells gene therapy. cell line gene therapy. Why? Because 20th chromosome is not a sex chromosome, not a germinal chromosome, it's a normal chromosome, somatic chromosome. So by somatic cell gene therapy, SID can be cured. What can be cured? SID can be cured. Sickle cell anemia, you might have studied because of the valine and glutamic acid, all those things, molecular biology you would have studied in the chapter. Sickle cell anemia can be cured. Hemophilia, which is known as Royal's disease. Royal's disease. Queen Victoria was suffering from this disease. Fine. So Royal's disease, hemophilia can be cured. Sickle cell anemia can be cured. SID can be cured by somatic gene therapy. By germinal gene therapy, one disease you might have studied in many first year chapters. Lung disorders, cystic fibrosis. This is also genetic disorder, genetic defect. You might have studied in the genetics chapter. Cystic fibrosis. Cystic fibrosis. This disease can be cured. Like this, similarly, many disorders, many genetic disorders, many genetic syndromes can be cured if detected at the embryonic level or at the very early stage level, or otherwise this therapy is also not possible. When a child is born with Down syndrome, Klinefelter syndrome, Turner syndrome, this question may come to students: Can these children can be cured? They cannot be cured after birth or before the birth or very immediately after the birth. Defective genes has to be replaced by normal healthy genes, then only this is possible. So this is the whole topic of gene therapy. Refer the study material, go through your textbooks, fine. Repeatedly watch this class. Do take very good care of yourself. Keep studying. God bless you. Good luck. Take care.